and thank you all. I think I actually went a little bit early then, so I'm going to restart. Firstly, I would like to welcome our audience and thank you all for joining us today for this webinar. My name is Sally Tucker, and I'm the President of Global Services for Lexitas. We are an ophthalmology-focused clinical research organization, and we have an integrated reading center that we're extremely proud of. I'm honored to be joined today by the leadership team of Voxeleron, our technology partner, as well as Allgenesis, a clinical stage biopharmaceutical company dedicated to the development of novel therapies for both the front and the back of the eye. We worked with Allgenesis on their recent phase 2A diabetic macular edema study, during which they also engaged with our reading center services. Today, we will have the opportunity to hear directly from Allgenesis on their exciting science and how they leveraged Lexitas Reading Center to support their clinical study. During the course of this evening's webinar, if you have any questions that you would like to submit, I encourage you to do that in the chat box, and we'll address those at the end of the event. But without further ado, I'd like to hand over to each of the panelists to introduce each one of them. So Tang, if I could ask you to go first to make your introductions.
Well, thank you, Sally. My name is Tan Nguyen. I'm the Vice President of Research and Development at Allgenesis Biotherapeutics. It's nice to meet you all.
And then Janet, would you like to go next?
Sure. Hello, everyone. I'm Janet Cheatham. I'm a clinical consultant with Allgenesis and have been working on this program for a couple of years. Welcome.
And Sunil? Oh, you're on mute.
Hello, this is Sunil Patel. I'm the Chief Medical Officer for Allgenesis, and really enjoyed working with Lexitas and the Reading Center to finish our phase 2A clinical trial.
And Daniel.
Hi, I'm Daniel Rusikoff, and I'm the Chief Technological Officer of Voxeleron.
Wonderful. Okay, so I'm going to now go over to some of the panel questions, and I'm going to address my first question to you, Tang. Perhaps you could tell us a bit about your asset and the journey that you have had to date.
Well, sure. A little bit more of the company. The company was founded back in 2014, so we're a small biopharmaceutical company. We are developing AG73305, which is our novel Fc fusion protein, and this is being developed having two domains to target two different pathways in retinal indications. And we're developing this for right now diabetic macular edema, and we plan to pursue this into other retinal indications such as neovascular AMD and RVO as well.
Great. So I don't know whether Janet or you, Sunil, want to address my next question, but what were the most important images that you collected during your recent phase 2A DME study?
Sure.
I would say that, go ahead, Janet, then I can follow up.
Okay,
Sure.
yeah, I'll start and then Sunil can add to that. So optical coherence tomography, OCT, is really the standard for the retina trials. And so that's probably the most important thing. And Voxeleron did all of the grading for this.
Okay.
And
Sunil,
you want to add?
Yeah, and I think the OCT and the quantification of the OCT is extremely valuable and important. Without it being standardized by a reading center, the numbers just really don't mean anything. So the OCT itself was very valuable, and then the volumetric analysis, in this case, the center subfield thickness, was the objective data that we can correlate that with the visual acuity. And we also saw the improvements on the CST with our molecule over time. And so we were able to correlate the objective data, which is the CST, center subfocal thickness, with the subjective data, which is the visual acuity. And we were able to formulate a plan for our molecule development for the next few trials. So it was extremely valuable in having the reading center give us some objective data on our patients and our clinical subjects.
Right.
And if I could just add something, when you're conducting an open labeled study, having a reading center grade all of those images really reduces any kind of potential for bias. And that really is a huge boost for the scientific integrity of your trial.
And so going on from that, what would you consider when choosing the reading center to partner with in your clinical trial?
So I think one of the things is really the responsiveness. How quick is the reading center to look at the images, making sure that they were collected per protocol, because you want to make sure that you can make corrections at the site level as quickly as possible. And then in some studies where you have to ensure that the patients meet a certain subfield thickness as an entry requirement, timeliness is everything. The other thing I think is the flexibility. I really want to have a reading center that is willing to work with us, particularly on an early phase study, and to contribute to, what are some of the maybe the intricacies of either the collection or some options for different images as well.
Yeah, and if I may add, I think the accuracy of the reads are extremely important, and also the consistency of the reads. Because if they're not consistent, you're going to add other variability, which is going to confound your data. So it's super important to have the accuracy and consistency to eliminate those variables. So I think Lexitas did, and the reading center provided us with that. So we had a pretty objective data that we can actually go through analysis.
Just the one other thing is that I think it is important for the sponsor to have access to those images. That's really important. Sometimes it's a matter of just understanding what is going on with an individual patient. Sometimes a sponsor is really going to need to have access to those images so that they can share it with other members within their organization.
And that's something you did have access to because of the iNebula software that you had access to as part of our partnership with you, which enabled you to have real-time access to the images. So I take it that had a massive impact in how you could use the data on an ongoing basis.
Yeah, I think for us, we're a small company, we're a startup company. Being able to access those images kind of gave us encouragement because we can see in real time the effect of this drug is happening for these patients, and so that brings excitement to us that our drug is working. But also, having those images available to us, in real time, being able to kind of, look through all the data gives us an opportunity to kind of present that information to our board of directors, kind of giving them an idea where we're going with this product and where we're going in the future with this product, and giving us that encouragement to move forward with the program.
And Daniel, do you have anything else that you want to add in relation to the iNebula software?
Absolutely. I mean, I hate to call it. Well, I hate to call it a game changer just because it's it's an overused term. So let's just say it alters the playing field, having access to the images. I think it helps to take a quick step back to ask why sponsors don't typically have access to their data, and it really comes from this interoperability problem in ophthalmology. I did my computer vision graduate work in radiology, and when I got into ophthalmology, I was shocked. I mean, shocked with the state of the data formats. Coming from radiology, everything's in DICOM. All the manufacturers, almost all the manufacturers, export in DICOM, which is an open imaging standard. And if you're not familiar with DICOM, as I imagine most people are, it's basically like JPEG for medical images. In ophthalmology, though, every device manufacturer outputs their images in their own special format, which requires some gymnastics to interpret. And it's for that reason that sponsors have gotten used to this this crazy paradigm that like the trial sites just throw their data over a wall to reading centers, and the sponsors often don't see it till the end of the trial. But it doesn't have to be that way, and it took a lot of effort. But we put the work in to understand all the formats, to where a site can just drag and drop their images directly to iNebula, and the sponsor can instantly see that data. And that's how it should be. It's kind of funny though, because we had some sort of unexpected headwinds as we fielded this application. Originally, when we first started, the sites came up to us and said, "Well, we're used to receiving these emails telling us that the data's up. How do we know the data's up?" And we were like, "Well, log in, go look at the images. You can see them; they're up." And it's crazy that was what they'd gotten used to. It's such a foreign concept. The idea is that the images are right there. We actually ended up having to implement that email notification also, because it makes the sites feel a little better. But sponsors have gotten used to this idea much faster, and they seem to really love having real-time access to the data. They've already weighed in here, but the norms here for Allgenesis, but the norms for most sponsors seem to be checking it multiple times a day.
Great. So I'm going to change track, and Tom, I'm going to ask you a question now. There is lots of discussion across our industry with the use of biomarkers, especially in posterior segment studies. I'd like to ask what your thoughts are surrounding this.
For a company or any kind of development company in drugs, I think biomarkers are going to be very important to kind of get an early gauge of how your drug is working in the patient population. In this regards, for the retina program, for DME, we know inflammation is a key component in the disease progression. So having some type of imaging or biomarker analysis on the inflammation is going to be very important for us. And that's something we wanted to look into for our clinical trial. And when we move forward and looking, to the future for other indications such as neovascular AMD, we believe that our molecule has antifibrotic activity, and it has a potential to regress the choroidal neovascularization in the AMD patient population. So having the ability to look at those type of biomarkers in imaging and whatnot, it's going to be very helpful for us to better understand how our molecule is working.
Yeah. And Sonal, I don't know if you want to, add to that and share kind of the role of the reading center and how they played a part in evaluating the endpoints that you determined for this study.
Well, I think the reading center was extremely helpful in us working with the endpoints. One of the endpoints obviously was our endpoints were BCVA, but the CST, center subfocal thickness, was very, very important to correlate, as I mentioned earlier, with the subjective data. It was the objective data that allowed us to correlate that. The other thing we did look at was OCTA, OCT angiography, to look for foveal ischemia and capillary density to see if there was improvement in the density. I think that would be extremely important going forward, because if we can regrow some of these vessels, we may actually not just stabilize the disease state, but may actually improve the disease state such that the patient may function better. I should also mention that the other biomarkers that are also becoming really important are some of the cytokine and growth factors. And so we are looking at that. It's been really important to correlate that with some of the imaging studies and objective imaging studies and how that correlates with the biomarkers. And so that we don't have to do biochemical analysis on all of these vitreous samples, but we can actually get an idea based on imaging what specific biomarkers may be going up or down.
Okay, thank you. So Lexitas partners with Voxeleron, which is our technology partner, and they provide cutting-edge AI automated technology in addition to biomarker identification. I think, Daniel, I'd like to pass over to you for you to share the capabilities and technologies that you have in this area and how they can support in clinical studies.
Sure, absolutely. Let me share my screen here. So it's cloud-based, so this is just a standard web browser. You log in, and immediately you can see all the trials that your account has access to. Each trial has a dashboard recording the key performance indicators for that trial. So, for example, here, recruitment of sites and enrollment of patients. You can also see the state of each visit. Workflows are fully customizable, but this example here shows a single read workflow. Um, this subject, for example, has 3 visits. The first is complete, the second one is being read, and the third has been uploaded and needs to go through image quality review. These workflow states can be filtered. So, for example, if you're just looking for a queried image, you can get there very quickly. Or, sorry, I should mention this is this demo is done on our staging environment, not our production environment, so it's not quite as responsive. You can quickly get to the queried images, and you can see the full workflow history for any given image. But now, so let's say I log out and I log in as a reader, and I want to grab an image to read. The reader accounts are similar to the PM accounts, but have slightly different sets of permissions. So I'll go here. I'll look for a readable image. Here's one, and I can either I can see this one's been read. I can either add to that read or establish a new read. In this case, I'll add to this read. And while it's downloading, this is a good time to mention that our software is developed under strict design control inside a quality management system that's been certified ISO 13485 compliant. So typically, sponsors have to choose between reading centers that are using, either validated OEM software but offer limited endpoints, or university software that's been pieced together and maintained by grad students and postdocs. So yes, they may have more endpoints, but they lack validation, and the software can be very clunky to use. So we offer the best of both worlds and have a large selection of advanced endpoints with an OEM level of design control and validation. So our AI-based algorithms are often all you need, but when they do fail, and trust me, they will fail. Anyone who tells you their algorithms work 100% of the time, or don't trust anyone who tells you their algorithms will work 100% of the time. So in the cases where the algorithms do fail, we offer 2 options. There's a simple per slice. Do this slice because this layer is more interesting. A simple per slice editing, where you can just play with each slice and see where you go. Or our patented intelligent editing system, which is designed to minimize your interaction with the system and propagate your edits throughout the volume. We also have standard measurements like calipers, freehand areas, and text annotations. And these can come from a vocabulary defined by your specific trial. We also have an integrated case report form, which auto-populates directly back up to iNebula. So while you're doing your read, it happens directly in line. So when you save this, all this information goes directly back to iNebula. So this is also a good time to mention. So then it goes back into the case report form. The case report form is designed at onboarding. Each CRF has different entries for different visits, capturing exactly what you need for each visit. And our system is fully 21 CFR Part 11 compliant. So we have a full audit trail of all interactions with the system. So let's see then what the sponsor sees. We made a lot of we had a lot of discussion earlier about the importance of real-time access to your data. Sponsor basically sees everything the PM sees, but they slightly less information. They don't need, for example, access to the full workflow. So they basically just see when an image has been received, when it's been approved as readable, and when it's complete. So you saw the Heidelberg in E2E earlier. We also support Carl Zeiss in their non-compliant DICOM format. We support the TopCon in FDA and FDS format, and OptiView in their OCT and then their RAW format. Sorry, and so let's see. So digging then back into biomarkers, log back in as a PM. We were very excited that that Allgenesis took advantage of several of our advanced biomarkers, including fluid volumes. So we delivered intraretinal, subretinal fluid volumes as well as PED. It can be delivered and shown in a variety of formats. So here's just a video overlay. We also offer image overlay as well as a full PDF report. We also offered them for this trial OCT angiography endpoints. So here it was foveal avascular zone, as well as capillary density. In addition, we've done a good deal of work. That's not all we offer. We've done a good deal of work in other biomarkers. So, for example, this is a paper published in the British Journal of Ophthalmology, where we validate our algorithm on more biomarkers. So instead of just IRF, SRF, and PED, we split PED into neovascular PED and drusenoid PED, and we also added SHRM, or subretinal hyperreflective material. The upshot of this study is that we offer expert level performance for all categories. In addition, we offer GA segmentation and progression analysis, and we're in the middle of a multi-center study here to validate it. These are some initial results. And we've done a good amount of work in study population enrichment, which is something that's been very sort of talked about these days. So, for example, here is a paper we published on predicting conversion from dry AMD to wet AMD. This is a paper being presented at this year's ASRS on predicting treatment response in diabetic macular edema to anti-VEGF treatment. In addition, we've done a number of other projects, specifically with Lexitas here on corneal confocal microscopy for nerve fiber quantification and a hill of vision analysis. So basically, the idea here is that we have a very robust R&D program, and if there's something you want and don't see, there's a good chance we already have it, or we're doing something similar, or that we have related technologies and could put something together very quickly. So please don't hesitate to get in touch. Image, or you can look at it as a report, a PDF report. And so here, this is just showing IRF and SRF. We did, this is just one of the reports. We did PED also for this study, and it's probably helpful just to bring up a few others, just in terms of AI-based endpoints. We did the 3 for Allgenesis there, but this is just showing other capabilities. This is a paper that we just published in the British Journal of Ophthalmology just this month, showing the IRF and the SRF from before, but PED now has sort of been divided into neovascular PED and drusenoid PED, and then we've added subretinal hyperreflective material, SHRM. And so we have an analysis there, where we show we achieve expert level results here. We also offer GA segmentation progression. This is part of a multi-center study that we're working on now, showing excellent results. In addition to some of those specific endpoints, we also have AI work in other sort of more cutting edge things like population enrichment. So, for example, this is work we've done looking at identifying fast progressors in AMD. This is work that we've looking at for DME, predicting response to anti-VEGF treatments. So there's a number of endpoints like that we offer. In addition, we've done some work with Lexitas on corneal confocal microscopy for nerve fiber quantification, hill of vision analysis. Basically, you name it, we have been working on it, and have really enjoyed our partnership with Lexitas, helping them provide these endpoints.
Thank you, Daniel. That was fascinating. So appreciate you sharing that with us all today. Before I move on, I'm going to just address a question that we've received. Other reading centers mentioned that they need an initial quality control step before the images are online. Is there a similar step for your software? And if yes, how long would it take for these two steps? One, immediately, or two, validated? And we have an initial quality review once images are received, before images can be read or graded. So these are manually checked after uploading. The time it takes is something that we negotiate with the sponsor, dependent on your needs. The image quality reviewer, if they find an issue, then it is highlighted to the site, and the visit is queried. But after it's read or graded, that's when the images become available for you to view. So we make sure that they are of a certain quality. I hope that answers your question. Moving on to post-hoc analysis, Janet, I'd like to ask you, what do you think the importance of post-hoc analysis is in clinical trial data?
So some of this depends on where in the clinical development your particular trial is. For early development, I think it offers you a lot of flexibility in terms of being able to do post hoc analyses, maybe even post hoc grading of a different endpoint that you hadn't previously considered. If this is phase 3, you'd be locked in and you wouldn't be able to do anything. But being able to do a post hoc analysis, particularly for a phase 2 study, can be extremely informative of what you are going to end up doing for your next trial.
Absolutely. Okay. And given what you know about the bioidentification capabilities that Daniel just overviewed, how might you partner with a reading center such as Lexitas in the future? I don't know if this is one for you, Tang, or for you, Janet, but both feel free to interject and provide comments.
Janet, you can go first.
Sure.
So on biomarkers, I think this goes towards, again, in an early phase study,
yeah,
there typically the focus is supposed to be on safety, right? But all the sponsors, all the investors really want to know about efficacy. And so that's where these biomarkers come in, because these studies, they're small, right? It's early phase. So they're small by nature, and you look at every single patient in quite a bit of detail. So sometimes I think these biomarkers can be really, really important. And besides the CST, maybe it's the intraretinal fluid or foveal transition zone or something like that is going to be important for your asset and the indication.
Yeah, I would echo that. Again, based on what you know about your mechanism of action of your molecule, you want to be able to look at some of these biomarkers to assess whether or not that you can get an early read on the efficacy of your molecule. Before you go into visual acuity or CST, you may be able to look at other biomarkers to kind of gauge how your molecule is working based on your mechanism of action.
Great. I'm now this is my final question to you all, and I think that I'd like to ask all of you, what lessons did you learn from your current program that you'll carry forward to your next study, and what advice would you give to other sponsors?
So for me, I think going through this trial and working with Lexitas and with Voxeleron, it was very useful and helpful for us to be able to see these images early and daily, as Daniel had mentioned, to kind of gauge what we're doing with our molecule, how to plan ahead with what we want to do for our next clinical trial, etc. So I think, lessons learned here is having the discussions with your CRO and the technology to better understand how the technology works so that you can better assess your endpoints in your clinical trial.
Thank you, Tan. Janet, Sunil, anything to add?
Yeah, I'll add to that. The other thing I would mention is going forward is extremely important, as mentioned by other guests, that it's important to actually have access to the data immediately, because you can make adjustments in the trial and look at some of the variables. And we did that with this trial. We had access to the OCTs, and we were looking at the central cysts and how it varied with different dosing of our molecule, along with time. And we were trying to see what the mechanism of the edema was, whether it be inflammatory or anti-VEGF mediated. And there's no way we could have done that analysis after the fact. It was extremely important to get that data right then and there so we could do analysis through that. So I think the system's terrific to be able to do that. And I think we did definitely have really good consistency in the reads on the CST. So on the longitudinal level, we can actually look at that over time and look at the real OCT and correlate with the CST, and correlated pretty well. And so I think overall, I think it's extremely valuable in carrying on a new study, and probably it's going to be really important in phase 3 as well. So I think we were very pleased with working with Lexitas and the imaging center to get the data we needed and do some analysis in the interim.
I'll just add, I didn't have any complaints. And for me to not have any complaints about a reading center, that's really kudos to them. So I like what they're doing and would like to use them again in the future. So you want to be able to select your CRO, your reading center that is the right fit for your trial. They're not all created equal. We need a lot of different reading centers out there, but you want to find the right one that is really going to work with you. I think there were some other questions that had popped up during the course of this little webinar. Yeah, one of them was on the OCT techs, and did we certify them? Yes, we did. And of course, you really have to make sure that you are registering the equipment, right, with the reading center. Some studies, particularly the larger studies where you might have hundreds of sites, you don't have the luxury of being able to require only one type of manufacture of OCT. You're going to have multiple different platforms. So you're going to have a reading center that can handle that. So that's important to know. Yes, we did collect other images. This is a DME study, so we did fluorescein angiographies. And that was something else that we did look at in these patients as we were going through, really trying to understand a little bit more about the particular mechanism maybe for these patients. So yes, certifying the sites, the technicians is important, and having that flexibility in the platform.
And sorry if I can interject briefly, that's another spot where having real-time access to the data helps, because you can turn around those certifications really quickly if the data is up there immediately. That only helps recruitment or site recruitment, and just speeds along the onboarding process.
Great. Well, I'd like to thank all of our panelists. So thank you, Tung, Janet, Sunil, and Daniel. It's been a pleasure talking with you today, hearing about your experience. I'd like to thank our audience for dialing in and listening to us all today. And for those of you that are ARVO, please feel free to come and say hello. We are at booth 4615. We'd love to see you, and we'd love to talk to you further about capabilities of our reading center.
And we also have a poster on our study. Ton, you want to give the details?
Yep.
Absolutely.
Yeah, we're presenting on Monday at Arvo. So we'll just look for Allgenesis. I can't remember our poster number, but yeah, we can provide that later.
Fantastic. Thank you, everybody, and thank you all.
Thank
Thank
you.
you.
Bye-bye.
Appreciate
Thank
it.
you.
Thank you. Bye.
Bye-bye.